I'm Nidhi Kumar and you're watching Science Time, a show that brings to you the best that science offers, from exciting developments in science and technology to futuristic solutions. Gap-free human genome sequence completed for the first time. Ozone may be heating the planet more than we realize. A new cancer treatment uses the body's cellular waste disposal to flag harmful proteins. And let's take you to story number 1. Well, this week, the Telomere to Telomere T2T consortium has published a complete human genome sequence, filling in gaps that had stubbornly persisted for over 20 years since the publication of the original Human Genome Project. The sequence is published in the journal Science. The first one, deciphered 20 years ago, included most of the regions that code for proteins but left out about 200 million bases. The rungs that make up the ladder of the famous double helix DNA structure, until now about 8% of the genome code, was missing from the blueprint. The newly determined sequence covers previously inaccessible regions of chromosomes, including all of the repeat heavy centers and short arms, and for the first time, scientists will be able to delve into the functions and variations of all 3.055 billion letters of DNA that code for a human. Most of the newly added DNA coding sit in the dense middle sections of each chromosome, the neat bundles of tightly coiled DNA found in the nucleus of almost every cell in our body, as well as the protective caps or ends called telomeres. The breakthrough was being able to interpret the very repetitive stretches of DNA sequences that occur in these regions. In the early 90s, when the first attempt at cracking the human genome began, the science of reading DNA was still somewhat limited. The repeats could too easily be skipped or their bases linked together incorrectly. Since then, technology has moved on, making it possible to accurately read all of the DNA, including the long parts containing rems of repeating code. The team behind the research describes a new era for genomics, where no region of the genome is beyond reach. The sequence contains previously undiscovered genes and previously inaccessible repetitive regions that can now be detangled. The work opens the door to complete end-to-end genome sequencing being possible for all species on Earth. The resource helps explain how to create every human cell in the body, which could shed light on how to treat, prevent, and cure disease. And with that, let's take you to story number 2. And ozone may be weakening, one of the Earth's most important cooling mechanisms, making it more significant greenhouse gas than previously thought, research has found. A new study has revealed that changes to ozone levels in the upper and lower atmosphere were responsible for almost a third of the warming seen in ocean waters bordering Antarctica in the second half of the 20th century. The deep and rapid warming in the Southern Ocean affects its role as one of the main regions for soaking up excess heat as the planet warms. The majority of this warming was the result of ozone increases in the lower atmosphere. Ozone, one of the main components of smog, is already hazardous as a pollutant, but the research shows it may also play a significant role in driving climate change in the coming years. The new research by an international team of scientists and led by the University of California, Riverside is published in Nature Climate Change. Dr. Michaela Heglin, associate professor in the atmospheric chemistry and one of the study's authors, said, and I quote, "Ozone close to Earth's surface is harmful to people and the environment, but this study reveals it has a big impact on the ocean's ability to absorb excess heat from the atmosphere." These findings are an eye-opener and hammer home the importance of regulating air pollution to prevent increased ozone levels and global temperatures rising further still, unquote. And with that, let's go to story number 3. And scientists from the Center for Protein Degradation at the Institute of Cancer Research in London are perfecting a new anti-cancer treatment that exploits the body's own cellular waste disposal system. Many modern cancer drugs work by blocking the function of the harmful proteins. Our cells have evolved a highly efficient technique for removing harmful proteins. It's a part of everyday business of life. However, our cells' protein degradation processes only recognize a specific number of proteins. Science has now found a way to add to that number, to put flags on new proteins that the cells wouldn't otherwise touch, but which we now realize are harmful and involved in tumor development. We are saying to ourselves, please can you add this harmful protein to the trash that you're taking out? An early example of this approach is provided by the protein degradation drug lenalidomide, which is used to treat blood cancer, myeloma, and which has been shown to improve patients' quality of life. And with this, we come to an end of this edition of Science Time. We'll be back with more exciting stories from the world of science next week. Until then, stay tuned to India Science. Thank you for watching. Namaskar.